So continuing in the same vein of this detection of microbes, we've got Dr. Evan Cheney. He's the Manager of Scientific Affairs for Roka Biosciences. Both Roka, well, all three of these folks have pods. So if you haven't seen them, please go visit them during the next coffee break. And I'm going to hand it over to Evan, and he's going to talk about the Roka Biosciences technologies.
All right. So it's a pleasure to be here at PMA and have the opportunity to talk about innovation and detection methods for foodborne pathogens, and to kind of show you how we as a company at Roka have tried to innovate that pathogen testing technology in a manner to provide a unique solution to some of the challenges that are faced when testing food matrices. So to give you a brief overview of how the little discussion will progress here, I want to talk briefly about microbiological methods in general and put those into a timeline perspective with regards to food safety. Then I'll talk a little bit about method selection criteria. So Roka is one company in this space. There are lots of things to consider if you are a company that has to send your product out for testing. There's characteristics that become very important in that decision. And then we'll talk briefly about the technology, how we've innovated, what our solution is, and how it differs from some of the other technologies. And then hopefully have some time to run through some data that's applicable to produce. So here's a slide. It's a bit busy. But when we really look at microbiological methods, specifically for detecting pathogens, it's really a young discipline. Around the 1700s, is really when the first prokaryotic cell was identified by Anton van Leeuwenhoek, who invented the microscope. Around 1800s, we have who I consider to be the grandfather of food microbiology, Louis Pasteur, who disproved spontaneous generation. Around that time, we also have Robert Koch, who developed Koch's postulates to say, if I have this bacteria and I put it into this animal model, and that animal becomes diseased, and then I can isolate that bacteria out, it's the causative agent. So that's what we have around that time. So it really wasn't until the early 1900s when we really started to understand the biochemistry and a lot of the properties of some of the important pathogens and were able to begin developing enrichment media and culture media that would help pull those pathogens out of a sample. And then of course, 1953, we have Watson and Crick and the description of the double-stranded DNA helix. In the 60s, we have our first ELISA-based methods, so our first antigen-antibody type methods, lateral flow, the pregnancy test type methods. That's where their technology was kind of the foundation began. And then 1983, when Kary Mullis developed PCR, which is largely the technology that we utilize today. So this is where it gets interesting. So 1993 is when we have the big E. coli 157 outbreak in ground beef, so Jack in the Box outbreak. And this really changed the shift of food safety. Shortly thereafter, we have HACCP implementation and HACCP processing. And around 95, 96 is when we see the first rapid detection PCR-based methods really being implemented and utilized for food safety testing. 1990 to 95, this detection method called TMA, which our technology is founded upon, was invented and patented. And then here's 2006. What happened in 2006 in the produce industry? The 157 spinach, right? So that really changed the focal shift to the produce industry. So really what this slide is to show is that, going back to like Jack's talk yesterday and the exponential growth of specific areas, pathogen detection is one of those areas, and we're seeing very rapid growth. And that's driven largely by public health data, CDC, FoodNet, so that tells us what pathogens are really important. That drives regulation, and then we have consumer groups like Stop Foodborne Illness, who also influence regulation and drive innovation in method technology. So we believe that we've innovated and are on the forefront of providing a unique solution. So I want to talk a little bit about our solution. But what's the big thing here? What happens when you have a need and there's a whole lot of innovation? It means you have a whole lot of choices, right? So in making those choices, especially if you're a producer and you have to have testing done on your product, it becomes really critical on how you define what method you're going to utilize to do that. And so some of the questions you may ask are, Who completes my testing? Am I going to test in-house, or am I going to send my samples to a third-party lab? What technologies are available to me? What technologies are available at that lab? What criteria do I seek? And what results are important to me? So these are just a few of the key things that must be considered that we found through interacting with our customers. And so in determining what that right method is, some of the key ones that we found are, of course, turnaround time. And this can be commodity specific, right? So produce industry, you've got a pretty short shelf life product. And of course, depending on what point in the production continuum that you're sampling, that the time to result may or may not be quite as critical if you're testing at the pre-harvest level as if you've got packaged finished goods. So culture methods obviously take a long time, 3 to 4 days. Antigen antibody-based assays can take 2 to 3 days. And most molecular methods, PCR and hours, you're looking at a 1 day to maybe a 2 day result for that. Once again, the technology is quite important for consideration. What is the accuracy of the result? What's the false positive rate? What's the false negative rate? But one that we find critically important is the matrix. Are you confident that the matrix you have to send off for testing, your product, that it does not interfere adversely with the efficacy of that detection technology? That's one big key consideration. Another key consideration is the method. Has it been validated independently? Has it been peer reviewed? Is it accredited and been compared to the gold standard reference method by AOAC, ISO? These are other key considerations. And so the overall confidence too is the manufacturer of that method. Do they follow GMPs? Are they certified by ISO? These are other key considerations. So that's just kind of an overview of what we found through interacting with our customers. Now, to get in the specifics of commodity characteristics and how they can influence a test, what we see in produce is one, which Dr. Lee, if you were at his talk yesterday, did a great job of describing. That is the natural microbial ecology of these different plants. They have an enormous amount of different background bacteria, and it can be diversely different between the different food products. And many of those can actually outcompete the pathogen that you may be testing for and looking for within an enrichment method. Chemical compounds. Many properties of plants can have bacteriostatic, bactericidal properties. So the enrichment method has to be able to deal with those so that you can grow up that organism. And then, of course, pigmentation. We see that some pigmentation can actually affect the efficacy of these different methods. So now I want to talk briefly about the innovative technology that Roka has developed and is offering to the food space, and then we'll try to go into a bit of data. So our technology is really founded upon 3 core technologies that all are a part of the method in one test. This starts with what we call target capture, and I will go into that on the next slide in more detail because it's really quite important for food testing. The second component is what we call transcription-mediated amplification, and so this is our amplification step. How this differs from PCR is that if you're trying to detect, let's say, salmonella, and you have a DNA-based target, gene target, that salmonella cell may have 1 chromosomal copy of DNA. The way that we differ is that that same cell may have up to 10,000 copies of the ribosomes in the cell. The ribosomes are the powerhouses of the cell for energy. Each one of those ribosomes has an RNA sequence in it. And so we're targeting that RNA sequence. So we're starting with much more target, which helps us reduce the time to result and also increases our analytical sensitivity. So then what we do is once we have separated this, and I'll go into that briefly, we then use a reverse transcriptase. We make the DNA copy, then we use an RNA polymerase, and we amplify the RNA product. And the last component of that is what we call hybridization protection assay. We use a chemiluminescent probe to go in and attach to that amplified product, and so if it's present, we get our detection reaction. But as I mentioned, the key consideration here for this technology that's really powerful is this target capture step. And so we've talked a bit about immunomagnetic separation, where you have the antigen-antibody interaction that are used with coated beads, and then you can remove the bacteria, clean up the solution. Somewhat similar, but a much different and more specific approach. We utilize these magnetic beads that have oligonucleotide primer specific to the RNA sequence of that pathogen. So then we can capture that, pull it over to the side, and then aspirate off this liquid. So we're removing inhibitors, we're removing pigmentation, we're removing background microfluoronucleic acid, and then we can go in and wash that and have a nice clean environment for that amplification and detection step. So once again, that translates to 3 levels of very specific reactions to get you that positive, negative result. Specific binding at this stage for the target capture. Then you have probes that bind a different specific region of that same nucleic acid for the amplification. And then finally you have this probe that once again binds another highly conserved specific region of that nucleic acid. So it's quite a robust method and very specific. The third part that we believe is very innovative for the detection technology industry that Roka has done is to automate these processes. And so going back to Jack's talk yesterday about the areas that are really, we're seeing exponential growth, and one of those was robotics. So we've gone in and put that detection technology onto a completely automated platform. And this confers obviously many benefits. One, you're reducing touch points, which leads to a higher and more confident and accurate result because you're reducing error in multiple handling steps. We're also improving lab workflow. So those of you with Lean Six Sigma minds, you know, that applies in the laboratory as well. It creates checks and balances, RFID tags, barcode tracking, all those are a component of our technology, and so that can all be integrated into a laboratory information management system. And another key characteristic, it doesn't require as high of a skill level technician to run it. So you can free up your highly skilled individuals to be able to go and utilize their minds and do other things while the test is being performed. And that has great benefits. So overall, the sample goes in the enrichment bag, and aliquot comes out into this tube. It's lysed, which makes the nucleic acid readily available and stable. That tube goes onto the instrument, and out comes the result about 3 hours later. Show you briefly some data where we've seen successful application in produce. One thing that my group does at Roka is we run these very small verification studies. So to give you an example, all of our assays have, we've been validated through AOAC for the detection of salmonella in romaine lettuce. Other leafy greens, such as spinach, iceberg lettuce, these types of things are not intrinsically greatly different from romaine lettuce. So we'd run these small studies to demonstrate that the matrix is not inhibitory to the method. And generally, they're just small 6 or more samples of inoculated, non-inoculated. They may be composite-sized samples. They may be 25-gram samples. It just depends on what we're trying to demonstrate there. So this is what the data kind of looks like. All of our non-inoculated samples are negative. All of our inoculated samples were detected positive at the 10-hour test point, 24-hour, and they're also culture confirmed by the reference method. The key I want to point out here is look at what our mean aerobic plate count was on this spinach, 3.2 times 10 to the 7th. We only put 8 CFU, 8 cells of salmonella into that prior to the enrichment. That's a lot of background competition there. So the enrichment method works. The analytical sensitivity gives us the time advantage, and we're able to detect those organisms. And of course, our AOAC validations are done at even lower inoculation levels. So we've done this on a variety of other produce matrices for multiple assays. This is just some data where we're showing that the matrix has no inhibitory characteristics. Sometimes our customers like to send us samples and we're blinded to them, and so we give them the results back and then they give us the results back and tell us what it was they submitted. So we've seen good application across melons and fruit and some other produce varieties as well. One other area that we've really tried to innovate and be ahead of the curve is in the enrichment process. So many methods for the different pathogens, you have to use different enrichment media that's specific to growing that pathogen up. So we've tried to develop our methods so that we can minimize a lot of that. And so this is just an example where our E. coli 0157 and non-0157 S-Tech assays are harmonized and they're validated through AOAC to be so with our salmonella assay. So you can take your produce sample, enrich one sample, and test for both 0157H7, non-0157 S-Tech, as well as salmonella from that same sample. And this study was just showing co-inoculation, where we put both organisms into the samples and then tested them and culture confirmed everything. Just some other examples of successful verification done by one of our customers for our Listeria assay, salmonella assay, 0157 and S-Tech. A variety of commodities here, but one thing that I will point out as well is soil and water. Pre-harvest testing is going to become increasingly important, I think, with FSMA regulation. We actually had the FDA did a study that was published last year and presented at IAFP, where they compared the Atlas method on spent irrigation water, sprout irrigation water, and compared many of the available commercial methods, and the Atlas did quite well in that study. So we have a full menu, Listeria monocytogenes, Listeria species, Salmonella, 0157, non-0157 S-Tech. They've all been AOAC validated. So to kind of give you a brief summary on what we've covered here, rapid detection of pathogens is really a quite young discipline, and there's a lot of innovation, there's a lot of technologies that are being made readily available, and there are a lot of really important considerations if you submit product for testing. Produce matrices are quite challenging. It's really important to understand how your product may influence the test method that you're utilizing. And to sum up the Roka portion, we've really tried to be innovative as a company to provide a unique solution for food testing by automating the process with our Atlas technology, and then having that high specificity of the detection, so that when you get a positive, you can be quite confident that you're making the correct decision on what you have to do with that product before it goes into commerce and potentially impacts public health. So if you have any questions or would like to come see the Atlas, we do have it in the Learning Lounge. And with that, I appreciate your attention, and we'll
Thank you very much.
take any questions.
Thanks, Evan.